So we're going to start in a few minutes. Just for the people who have signed up early, I just want to do a little advertising here. You're seeing the thing for today's talk, which is Gavin. Just want to remind you in 2 weeks, we'll also have Shilpa talking on July 2nd. This is on this SRF Facebook page. You can register with that link at the bottom. She'll be talking about some of her specifically Syngap 1 epilepsy research. Hey, Gavin, you're logged in as Marta, but it's all good. And then also Helen Badeup will be 2 weeks after that. And then, so Gavin, I'm just doing a little general FYIs before we get going. A reminder that we have 5 days left on this Be Kind campaign. So if you want to get shirts, again, the link's on our Facebook, but SRF Kindness Off Bonfire. SRF does a weekly parents meetup, which is always fun and generally useful. I took the least flattering picture possible of everybody in this one, but they're good meetings. And then just a reminder, we have the Syngap Research Fund board is now up to 12 families, and we're always looking for people who want to help. So if you are, if you have the time to do more and you want to be involved, reach out. SRF is pretty much everywhere. We're on Facebook, Instagram, LinkedIn, Twitter, and syngapresearchfund.org is a good website to have handy. So with that, just in a minute, I'm gonna ask Marta Dehia to introduce Gavin Rumbaugh, and then we're gonna give it to Gavin. Gavin, can you, I'll just wait 1 more minute, and then I'm gonna ask you to take over screen sharing to make sure you can do it. Is that cool?
Sure.
All right, fabulous. And then I'm gonna, just in case anyone's worried, when Gavin starts talking, I'm gonna force mute everybody. And then at the end of Gavin's talking, I will unmute everybody. So if you have questions or concerns or things you wanna say that before we get unmuted, you can just chat them to me. So in the chat function on Zoom, you can chat any questions you want, either to me directly or to everybody, which would probably be most efficient. So I hear a lot of people chiming in. Let's just give it another 30 seconds. Reminder, you're here, Gavin's talking, Shilpa will be talking, Dr. Kadam will be talking, I should say, in 2 weeks about Syngap epilepsy. Dr. Badeup from Berkeley will be talking about some of her research. We are selling Be Kind gear. Feel free to get some. We have a happy hour every week, Wednesdays at 5 Pacific. It's a really good chance to talk to other Syngap parents. Sorry about the picture, Peter. I see you smirking. And it's fair. And speaking of Peter, there are 12 families on the SRF board right now. We are always taking more help. Hi, Dr. Terry Joe. Thanks for coming. And SRF is on Facebook, Instagram, LinkedIn, and Twitter. We try to push our content out on all of those things. So please follow us and stay connected. Getresearchfund.org. Ooh, so many cool people on the call. I am thrilled. So time is precious, and we're all going to run out of time because Gavin is very popular and we'll have lots of questions for him. So let me now hand over the screen sharing to Dr. Rumbaugh, and let's make sure he can do that. And then as soon as he takes control, Marta, if you want to, or actually Marta, why don't you just start talking and give your introduction? I know you did your research on this one. Marta, are you there? Yep, okay, you're in, Gavin, you're good. Marta, yeah. We can't hear you.
You have her muted, or she's muted.
Marta?
I'm mute.
Okay, there you are. Now okay. We can hear you. Please introduce Dr. Rumbaugh.
Yes, then I want to welcome everybody to the meeting. My name is Marta Dehia. I'm a Syngap parent, like everybody knows, and we are excited to start the Syngap Research Fund webinar. The goal of the webinars are to get everybody closer to science of Syngap, make you aware of the research being done and the opportunities to participate, empower your communications with clinicians as you get clearer knowledge of Syngap. As a group, we firmly believe will be able to improve the quality of life of our Syngapians, find the right treatment, and ultimately the cure. If we continue following the science. I want to tell you again, the name of the talk today is Syngap Research Roadmap, behind the scenes view on efforts to develop therapies. I want to I have the pleasure to introduce Dr. Gavin Rumbaugh, who is a neuroscientist at the Scripps Research Institute in Jupiter, Florida. Dr. Rumbaugh's laboratory is devoted to the understanding of how synapse and circuit mechanisms contribute to cognition and behavioral adaptations. In particular, he studies how genes that mutated cause a defined neurodevelopmental disorders like Syngap. Dr. Rumbaugh has written at least a dozen of papers on Syngap, including one that gives us reason to believe that our kids can be helped by a therapeutic agent. His laboratory was able to improve function related to seizures and well as restoring Syngap protein in an adult animal. Dr. Rumbaugh worked in his earlier years in a basic science of Syngap protein function, even before Syngap1 related disorders were identified in humans. He experienced a powerful change in his career after meeting his first Syngap1 patient, who motivated him to dedicate his efforts to find pharmacotherapeutics for this disease. The two main focus of his research are finding safe and effective therapeutics and creating animal models to understand behaviors to find effective treatments. Last, what makes Dr. Rumbaugh even more special is that he dedicates his work to make a difference in our kids' lives. And this is the introduction for Dr. Rumbaugh. And for people that just came to the webinar, again, this is Dr. Rumbaugh, and the name of the talk is Syngap Research Roadmap, behind the scenes view on efforts to develop therapies. We have the chat. In the chat, you guys can formulate questions, and we are going to have Dr. Rumbaugh give us time by the end to answer those questions. And for the people who could not be on this webinar, we are taping the webinar, and it will be launched later on. And go ahead, Dr. Rumbaugh. Thank you.
Oh, thank you for the wonderful introduction. Can everyone hear me okay? Yeah?
Yes.
Yeah.
And just quickly, Gavin, I'm going to force mute everybody. So, and then I'll unmute you. So just give me 1 second to do that. Okay. And then unmute. Okay, Gavin, it's all you.
And can you see my presentation and presenter in
Yes.
the main view? You can, and you saw the transition there. Yeah.
Yep, we're good.
And am I looking in the camera?
We can't see your face. We can just see your screen.
Okay, great. And I can't see you either, the way it's set up. So, you know, this is the days we live in. So it's a little bit different. So I'm going to try to give the same style of talk I normally give if I was there in person. But of course, we're limited because we're not, you know, physically next to each other. And so when I give talks to families and stakeholders, to non you know, what I call hardcore science talks, it's okay to interrupt me. And Mike has given very clear instructions to the audience, and I think on Zoom that works best. So Mike, if you feel like it's a good time to interrupt me with a point of clarification, please go ahead and do that. You know, I'm a big supporter of that style. So, and I understand that you guys were shooting for about 30 minutes, you know, of information that comes from me, and then maybe, you know, another 30 minutes of Q&A. So that's kind of what I planned for, but sometimes it's a little more organic, so it's okay to go that way too. Okay, so I'll start with should start with an outline. And so, but before I go into the outline, you know, I think it's important that you know, that you all understand that I'm here for you. So, you know, for this hour, you know, my time is yours, and we should use it in the way that's best for you all. And so the reason why I do this is because, you know, I've gotten tremendous amount of motivation and inspiration from working with Syngap 1 families and patients. So it's not just for many scientists, it's kind of an academic exercise. You know, we try to solve problems, but you know, it's become, you know, somewhat personal for me over the years. So, you know, I really want everyone to know that this is something that I care about very deeply. So, you know, my goal is to help parents figure out the best way to use their voice for helping their kids. So, okay. So maybe 4 parts of the talk. So how do genetic variants known to cause developmental disorders impact brain function? So what is the current state of understanding of what Syngap 1 variants do to the brain? So that'll be maybe 5 or 10 minutes. Sorry about that. And so the next part of the talk will be how do we group Syngap 1 patients? So we don't necessarily see all the Syngap 1 kids the same way biologically, and there's an important reason why we do that. And so we will talk a little bit about that. And then the last part of the talk is going to be on strategies for treating Syngap 1. Now, this isn't just my preferred strategies. I'm going to try to give you a synopsis of the kind of consensus of the academic labs and biopharma, so commercial entities, on the most promising and fastest routes to getting treatments for Syngap 1, and then also some more longer term visions to optimize and to come up with alternative therapies for Syngap 1. And then of course the Q&A. And again, Mike, feel free to interrupt me at any time. Okay, so what do we know about Syngap 1 disorders? So, you know, I'm trying, my job when I communicate to the public is to take the science out of the science and to just try to get the ideas across. And the reason why I do that is, you know, I think hard on how to communicate these complex ideas to people who aren't trained neuroscientists. And so the way that I approach this is, you know, if my nephew had Syngap 1 disorder, how would I explain and how would I raise the most critical issues, you know, to my brother or sister? And so that's what I'm going to try to do. So I'm trying to imagine that you know, that my child's affected. So what are the key things that you need to understand? Because there's so much information out there. There's more information than even I can assimilate, right? Or anyone, any scientist can assimilate. So you have to be able to filter and pick out the most important things for the problems that you want to solve. And so what I'm going to try to do is distill the most critical issues and concepts that as parents that are interested in helping to accelerate treatments, you know, what you should try to pick out and what you should try to focus on. So when it comes to the actual biology of Syngap, you need to know very little. And so, you know, you just need to understand that Syngap 1 is a gene, and genes encode proteins. So can you see my cursor, Mike?
No.
You can't see my cursor. All right, let me try. That's too bad. Let me try to go back here. All right, let's try this again. How about now? Can you see it now?
Yes, yes.
Okay, all right, great. So what you need to understand is that Syngap 1 is a gene, and so this is the gene here. And of course, you know, a lot of this you guys already know, but I just want to put it into a complete story. So Syngap 1 is a gene, and genes encode proteins, okay? And these proteins are, there are many different proteins. There are 20,000 genes, and each gene makes basically a protein, but those proteins can come in different forms and different flavors. And those proteins assemble inside of cells they assemble into machines, and those machines do work. And that's what carries out life. The business end of life inside of cells are proteins. And so in the brain, which is the most complex organ in the body, the brain cells are neurons, and their supporting cells, glia, assemble and connect to each other. And those create circuits and networks. And those networks are where memory, planning, behavior, everything that we can do, everything our brains can do, they can do because neurons to connect to each other and create brain circuits. So you can see this would be one synaptic connection. And the human brain has billions of neurons and trillions of synaptic connections, and right, and this is what you end up with is a complicated biological wired machine. Okay, and so what happens essentially is that Syngap1 gene encodes Syngap1 protein, and that's important for the way that these neural networks are assembled in development, and how they change with experience. So what do I mean by that? So first thing to think about is that these are the connections, and Syngap lives inside of these neurons, right? And so as the brain develops, these connections are formed. You know, we're born with very few brain circuits, and as we develop, we get more of these circuits and more of these networks, and then our capabilities improve throughout life. And so the big unknown in general in any genetic disorder, but especially in Syngap is that how does disrupted Syngap1 function affect the way that neurons connect and create networks, right? Because what the end result is, and what you really need to understand is that the loss of Syngap protein results in an alteration in the way that these networks form, and that is what causes altered memory, planning, language, motor function, sensory function, behavior, seizures, everything that you can associate with Syngap1 disorders. And so what's the consensus of the Syngap1 biology is over the past 10 to 12 years, a lot of it, I could say that my lab is driven is to try to understand when you lose Syngap1, or at least one copy of Syngap1, the closest approximation in an animal model we can have to try to understand brain function in rodents and how it may be affecting human brain function, is that Syngap1 or loss of Syngap1 does two basic things, at least two basic things. And one of those things is it affects plasticity. So the way that you store memory, the way that you learn and store memories, is that these connections here can change with experience. So when you learn something, you actually can physically make new connections, or existing connections can grow bigger in size, and then those connections become stronger. And that's one way that this organic neural network or supercomputer can store information. Okay, and so Syngap1 or Syngap protein that comes from the Syngap1 gene is critically important for those processes that allow synapses to grow and shrink, to store and erase information. Okay, and so the other thing that Syngap1 does is during development, it helps to facilitate the growth and maturation of developing neurons, and that's initially we're born and our brains are small, and it's because we have very few connections. And as our body grows, our brains grow, and as they grow, they develop more and more connections. And that's why you're in first grade you have limited capabilities, and by the time you get to high school, you can do calculus. And a lot of that's because your brain just has more processing power because it has more connections. Over here. And so what Syngap does is it's a completely separate job from this plasticity job. It's a role that we can differentiate. They're completely different functions of the gene, and it's to facilitate this wiring. Okay. Now, as Marta alluded to, I got a series of questions that were posed in anticipation of this talk, and one of them, and rightfully so, is that to what extent can deficits in a Syngap1 brain be rescued after childhood? And so we recently shown a couple of years ago now that these plasticity mechanisms, or the mechanisms in the brain that allow you to learn and remember and store information, those appear to be non-developmental functions of Syngap, meaning that they're used in development, but they're also used in the adult brain, meaning that if you can rescue the Syngap1 gene or fix the Syngap1 gene, that you may be able to recover some of this plasticity. And we have another paper that's been uploaded to bioRxiv that will be published soon, where we show that this plasticity happens at the level of what are called cellular ensembles, and these cellular ensembles are what actually store memories. And we show that these are disrupted in Syngap1 haploinsufficiency, and that these ensembles can be restored, and this plasticity amongst cells can be restored when you put Syngap1 back into the brain. So we're very excited about that. And we think that those capabilities don't depend necessarily on what are called developmental critical periods, and therefore therapies, these Syngap1 therapies could be beneficial even after childhood. Okay, so that's kind of wraps up the Syngap1 what we know about Syngap1 biology, and now I want to transition to the variants. And so, and a lot of this talk has been driven by conversations I've had with many parents, some of you that are on this call, as well as people in the biotech industry, biopharma industry, and other scientists. And so I think it's important to discuss the different types of variants and what they mean for thinking about therapeutics. Excuse me. So there's really two kinds of variants. We can put variants in two different kinds of buckets, if you will. There's the clearly pathogenic variants here, and what are called the VUS, or variants of unknown significance. And so the clearly pathogenic variants are the ones that have received the most attention over the years, is because, as you'll see, is that they may, while they may be different individually in terms of how they actually affect the Syngap1 gene at the level of the bases or the level of the Syngap1 gene structure, they're biologically similar or the same in a lot of ways. And so these clearly pathogenic variants or nonsense variants, or what are called base changes that lead to premature stop codons, small indels, large deletions. And what these types of variants have in common is that they essentially cause what's called a frame shift inside of Syngap1, and that will inactivate one copy of the gene. And we'll go into this a little bit more and why that's important. So the variants of unknown significance and some of you may have seen these on your genetic reports. And so these unknown variants are typically missense variants, where you have an amino acid change inside of Syngap. So you don't necessarily destroy the function of the copy of Syngap. What you do is you change the amino acid or the individual building blocks that go into the protein. You change one of those, and so often what that can do is destroy the protein. So the protein's made and it becomes unstable and it gets destroyed. But sometimes it doesn't do anything. And sometimes it can actually create a protein with a different function. So because of that, and the only way to differentiate those three possibilities is to actually do experiments in the laboratory to determine how the base change affects Syngap function. Whereas in the case of the clearly pathogenic variants, because you have what's called a frame shift and you have early termination of Syngap1 reading, basically we know that those proteins never get made and the mRNAs get destroyed. So we can group them into the same category. And so here you can see, I expand on this idea a little bit more, is here's a map of all the known nonsense variants in the Syngap1 patient population. A colleague of mine, Al George, has recently put this together because we're getting ready to undertake an exciting collaboration on how to fix these nonsense, potentially how to fix these nonsense variants, first in animal models and maybe someday in patients. And so the concept that I want you to take away from this is that while all of these are different nonsense variants here on this map, okay, that means they're all premature stop codons. And they all have different individual, and all your genetic reports, or by and large, all the genetic reports will say something different in terms of the base affected. They're biologically all the same in that they cause genetic haploinsufficiency, meaning that you have two copies of Syngap, but one of them doesn't work. So you're only left with one copy of Syngap, okay? And as I mentioned, these things are less clear for the variants of unknown significance, and where you would have to actually do research on every variant to know how the missense variant or even a splice variant. So a splice variant would be, or a splice mutation would be in the same category. And you have to do experiments on these individual variants or mutations to figure out what they're doing. And to some extent, that's being done. I know that some of us are working on these types of missense or splice variants to understand exactly what they do. Now, so why is this important? And so why is it so important? Why do people focus on haploinsufficiency? Why do people focus on these clearly pathogenic variants? And what do the families that don't have these variants, what can they take away from this? And what are they supposed to think? Because I get these questions a lot. So the reason why haploinsufficiency is so important and why it's such an important concept for all parents, as well as clinicians and biologists, is because it's the glue that keeps us together. So the unique genetics of Syngap, and one of the things that's very, very important is it's what's called an autosomal dominant disorder, and where you have what's called genetic haploinsufficiency in the majority of the patients. And you can think about, and I get this question a lot, and I think sometimes it's difficult to comprehend exactly what we mean by this. So I spent some time this morning and came up with 3 slides to try to show this graphically. So what exactly does haploinsufficiency mean? Why is it important? And how can you visualize it? So you can see these 3 panels here. Follow the blue arrow. So you have a genome, you have lots of chromosomes, and inside of all those chromosomes, you have your 20,000 genes. Within one chromosome, you can unravel it like a spool of yarn. And you can see that there are genes in green, okay? And so like this would be Syngap1, and this could be PSD95, and this could be some other gene here, okay? And so when they unravel, they can be translated. So basically what that means is that the Syngap1 gene is then made into message, which can be seen here. And you make this message, okay? And then the message can be, so that's transcribed in a message, and that can be translated into protein, Syngap protein. That's the part of the Syngap1 gene that actually does something inside of cells, okay? And so that protein, I think the best way to think about protein inside of cells is the way that cells work is they're made up of a bunch of large protein complexes. And what protein complexes are is think of each protein as a gear. And think of any complex machine. You have a lot of gears that were put together, and the sum of the parts is what equals the machine. Okay, so that's the best way to think about protein. So Syngap is just one gear, one part of a large machine that comes together, okay? But like any complex machine, you have some parts that are absolutely essential and some parts that, you can get away with not having that part work. Like imagine your car. So you have windshield wipers, right? And so you only really need windshield wipers, like in Florida, you need them a lot, but in a lot of places you don't need them very often, and you can even get away if one windshield wiper isn't working, right? So you can imagine like these green gears here are proteins that are made from the genome. And so maybe they're a little less important to the overall function of a cell or a neuron. But this red one here, if you imagine that this is part of a large complex, you can see that this gear here is important to turn all the other gears. And so if this gear doesn't work right, you can imagine that the whole complex machine doesn't work right. And so if you take this, what's called protein complex of a bunch of machines, and you have many of these complexes, and they fit together in an even more complex machine. And so this machine will turn all these gears, and then it'll turn this one, and it'll turn this one, and this is your flow of energy, okay? Now, so what happens in haploinsufficiency? So this is your Syngap1 gears, and in a normal neurotypical genome or person, you have 2 copies of the Syngap1 gene. So any gene that's not on the X chromosome, X or Y chromosome, you have 2 copies, one from mom, one from dad. And so each gene is functional. Each gene copy is functional. So, mom's copy of Syngap1 makes Syngap1 mRNA. Dad's copy of Syngap1 makes Syngap1 mRNA. And both of those mRNAs make proteins, and you get basically 2x of the protein. But in haploinsufficiency, because of the de novo variant that happens, basically when, a conception or shortly after, most of the time, you have that variant land in Syngap that destroys one copy, and it can no longer make mRNA. And so what you end up with is half of the Syngap1 protein, okay? So the neurons only make half of the number of Syngap1 gears. So why is that bad? Syngap1 is one of those proteins, or one of those gears that's essential for the way that neurons work. So it's not the windshield wiper, it's the transmission or the radiator, something that's fundamental that you can't really live with if you don't have enough of it. So you can imagine that this is this complex machine that I showed you, that this is the critical gear, and it turns all these other gears, and then it turns this gear, and then you can see the energy flow this way, okay? But when you have Syngap1 haploinsufficiency, this is where Syngap fits. But you only have half as much Syngap, but you have all the correct number of all the other proteins. So the machine assembles, but you don't have Syngap here to help turn the energy, and what you end up with is a breakage or a disruption in the flow of energy or the function of the cell, okay? So what we really need to do is to get these Syngap proteins back, because Syngap does more than one thing. It doesn't just do one job, it does many jobs. And that was the point of my first 3 slides, is that it not only affects plasticity or the way that brains change with experience, but it also changes the way that they organize in development. Okay, so this leads into the approaches to giving back Syngap1 protein. So haploinsufficiency is 80% of the known Syngap1 patients Syngap kids are thought to have haploinsufficiency, meaning that they, the root cause of their brain disorder is that they have low levels of functional Syngap1 protein, and that's what causes their disease. And so what nearly everybody is focusing on currently, and for good reason, is to try to figure out ways to bring these levels back to normal. And that's some of the work that I did in mouse models, is to show that when you put the protein back in using genetic tricks that only work in mice, but it's proof of principle that if you can put the protein back in, even after development, you can restore function. So now people are trying to take known biological mechanisms and to try to exploit them to try to trick the cell that only has one copy of Syngap and makes half the protein, to trick it into making twice as much Syngap. And so the idea would be, now you give all the gears back and now you can recover function of the neuron, okay? And so there are a couple of strategies. And so there's a couple of strategies related to giving the cell back its amount of Syngap, okay? And so plan A is what you've probably heard a lot about, and rightfully so, are ASOs, or antisense oligonucleotides. And so these are synthetic RNA or DNAs, basically, that trick one copy of Syngap into making either twice as much mRNA message, or to make the message that's there, to make twice as much protein out of it. Okay, and there's a lot of interest in ASOs, because these ASOs have been successful in raising protein in other genetic disorders, most notably spinal muscular atrophy and the ASO nusinersen. And so there are academic labs that are investigating ways to exploit ASOs to raise Syngap protein, and there's also commercial entities, several commercial entities that are exploring ASOs to regulate Syngap. Okay, so what the point I'm trying to get across, and this is what my lab works on, is ASOs may or may not work. And we have to be honest with that. It's experimental. ASOs may work for Syngap, and they may work for some kids, and they may not work for others. I mean, think of epilepsy medications, or even medications that treat depression. So 60% of patients don't respond to depression medications, right? And only a certain proportion of patients that have any type of epilepsy respond to epilepsy medication. So there's no reason to think that every Syngap child would respond to an ASO if they do work. And I think that they probably will work, but the question is, will they be able to penetrate the brain areas that need Syngap expression? How well will they work across the broad Syngap1 patient population? So because of this, and in any disease, I mean, think about blood pressure medications. So there are many different ways to treat blood pressure. And so there's usually, and the reason is because between the side effect profile and the different types of biology for each patient, you need more than one therapy. You need more than one arrow in your quiver to be able to attack disease. So what's plan B after ASOs? And the issue is that they're to raise protein expression. And the fact is, there really is no plan B right now. And so what YLab is trying to do is to find these alternative plans to be able to raise Syngap expression. So one thing I'm not going to talk about are alternative strategies to raising Syngap expression, and those would be, repurposed drugs that alter the RAS pathway, like statins and other related drugs, because at the end of the day, those don't treat the root. So those are interesting strategies, and they're important, but they're probably not going to work as well as therapies that raise Syngap protein expression. And the reason why is because the Syngap protein has many jobs. And so it's difficult to imagine that a drug that works downstream of Syngap function could fix all of the things that Syngap1 does. So what a lot of us have agreed on in the community, in the scientific community, is that the best way to attack a disorder where you have low Syngap, where you have low protein, is to try to put the protein back. And so that's why there's so much emphasis on that. And there's just two or three more slides. So what we hypothesize, and we know this from just neurobiological principles and neuronal cell biology, is that there's probably lots of ways to go from the Syngap1 gene to the Syngap1 protein. And so there are likely many different biological pathways that regulate ultimately the amount of Syngap protein that's in a cell. So you can make more message. You can make each message make more Syngap. You can make the message more stable and load more of that message into the ribosome to make more protein. And also we know from neurons is that each Syngap protein or any protein in the cell doesn't live forever. So most proteins only live in the cell for 24 to 36 hours. And so these proteins degrade. So, and they degrade through biological mechanisms. So if you were able to regulate the way that they degrade, you may be also able to raise Syngap protein expression. The issue is that we don't know anything about how Syngap1 is made or how Syngap1 is degraded. So we literally don't know any of the biology. So for some genes, we do know some things, but for Syngap, we don't know anything. And so one of the things that I've been trying to encourage people in the community, especially parent organizations that have interest in funding research, is to try to find people and support people that want to understand how Syngap1, how the Syngap1 gene is regulated, how the protein is made and how the protein is degraded, because those can lead to what are called druggable pathways that might lead to drugs that raise Syngap protein. So what does my lab do? Well, my lab, because we don't know anything, and so there's a trick that you can do in biology that if you don't know anything, you can do what's called an unbiased screen. And what that is that you say, okay, well, I think that you can regulate Syngap protein, but I don't know how. So what you can do is invent a system that shows you how much Syngap is present in a cell. And so what we did is we created, using our tricks and our tools for mouse genetics, is we created a series of mouse lines that can, every time a Syngap1 protein is made, and actually that Syngap protein that's present in a cell can make light. And so, so neurons will glow when they have Syngap in them, because the Syngap proteins themselves glow. And the amount of glow that is in the cell is directly proportional to the amount of Syngaps present. And this type of glowing, if you will, is very easy to measure across many tens of thousands of neuronal, what are called neuronal culture wells. So we have many neuronal networks. And because you can screen many thousands of them, if you can put different compounds in all of these different wells, the idea is that if you screen enough of these compounds, you're going to find that compound that does what you want, which is to make the cells glow more, which is to make more Syngap. So, but how can you do that? So it's not that hard, frankly, to make these systems that tell you how much Syngap is present. But what is hard is to screen the tens of thousands of assay wells. And so that's what makes Scripps very unique. So Scripps has 13 FDA-approved drugs. And one of the reasons why there's 13 FDA-approved drugs that came from Scripps is because we have this ability to screen tens of thousands of compounds across many assay wells using these high throughput robotics that you see here in this video. And these robotics are basically what you would see at Merck or any other large pharma, but we have these at Scripps in Scripps, Florida. And what that allows is researchers like me to work with experts in high throughput automation to figure out new ways to look for drugs. And one of those methods that we're using is to use this Syngap glowing assay to try to find the probes or compounds that raise Syngap protein. So that's something that wasn't able to be done in the past, but because of working using our understanding of the Syngap1 disorder and our knowledge of mouse genetics and the knowledge of the Scripps Screening Center, we're now able to do this type of high throughput screening to try to find those compounds that raise Syngap expression. And so I want to caution you that this is a longer term solution to discover non-obvious ways to manipulate Syngap expression. So the ASO pathways work, and Syngap1 is in theory one of those genes and proteins that can be regulated by ASOs. But again, there are probably dozens of different ways to regulate Syngap protein. And so one of the ways to do this is to find these compounds. And then once we find the compounds that can raise Syngap protein, our colleagues at Scripps have this amazing ability to work backwards from the compound to figure out the biological mechanism that made Syngap go up. And then the idea is that those pathways that we find, starting from that compound that was from the high throughput screen, we think that those will be the pathways that will be used to raise Syngap protein in the future. So with that, I just want to give a few acknowledgements. So of course, the screening facility at Scripps is run by Tim Spicer and Louis Scampavia. I've been working with them for 10 years. They were gracious enough to listen to my ideas and to think outside the box, because most screening facilities don't screen in neurons and have no interest, but these guys wanted to tackle this problem, and I think it's going to bear fruit. Also, the NIH has been instrumental in keeping my lab going over the last 12 years. They've been very generous in supporting my laboratory. Scripps has given me tremendous support. Also, various parent-driven advocacy foundations, SRF, Leon and Friends, and Bridge the Gap are game changers, I think, in the way that parents can interact with researchers to really drive research forward. And I encourage everybody to keep doing what they're doing, because it really is having a real impact, because you've only been at it for a few years now, and you know, imagine where we can be in a few more years. So thank you very much.
Awesome, Gavin. Thank you very much. I just got one question that came through, so I'm going to hit you with that right off the top.
Sure.
The question was about the neurons you're using in your high throughput screen. Are you using human neurons or other? Do you want to clarify that, since you are the mouse guy?
Yeah, well, so I'm going to stop sharing my screen. We can use both. So we have glowing neurons from both human iPS cells and from mouse. And so, you know, if you want a science explanation, you can read. We have a paper on this, and I can send you the reference. But to make a long story short, is that human neurons, what's the difference between human development and mouse development, right? At the level of neurons, their neurons are basically the same, honestly. What's different is the number of neurons in the way they connect in the brain, right? But the mouse brain and the human brain are very similar, okay? It's just the scale's different. But what's the difference? So a mouse gestation is 21 days. And its development they're sexually mature within 6 weeks, okay? A human gestation period is 9 months, and sexual maturity happens, 12 years of age or whatever. And so when you model neurons in a dish, you have to keep human neurons growing much longer before you get synapses where Syngap lives. We're talking a couple of months to be able to see real synapse biology, okay? Whereas in a mouse neuron, you can see just 12 days. You can start to see the type of biology that you need, the glowing that you need. And so the difference between 12 days and 2 to 3 months is millions of dollars in reagent costs. Right. So the idea would be, if we used human neurons, we could, and we will, and I'll get at that in a second, but you would be able to screen far fewer compounds, an order of magnitude fewer compounds. And so in order for high throughput screening to work, you have to look at, we're planning on looking at 200,000 compounds, right? Yeah. So 0.2 million. And you need that because they're rare. It's a needle in the haystack. And in order for it to work, you have to screen tens and tens of thousands of compounds. And what we're going to do is use the human neurons to validate. So the step one is to use mouse neurons, because we can go high throughput.
Awesome.
And then what we'll do is use the human neurons to validate. We'll prioritize the candidates that we find in mice that also work in human neurons.
Yeah, and I would point out, you've been very humble in this talk about how little you know. I mean, you are the Syngap mouse guy. So having you use your mice, which is, when other researchers call us and say they want to get involved, we're like, we'll call Gavin, get his mice. That's what everyone does. So it's what you're doing there is pretty awesome. And you mentioned the patient advocacy groups. So I would be remiss if I didn't do some advertising. Syngap Research Fund does have an SRF fellow in your lab, right? We've been funding someone for 2 years, and we're on board to keep that up for at least another year. So the idea is that hopefully we've beefed up your team and you guys can move even faster in spite of COVID. So my question stream is filling up fast. Heather, I've unmuted you if you want to ask your question, and then I'll go to Hans next. Heather, are you there? Oh, you've remuted yourself. This mute game is tricky. There you are, Heather. Go.
Can you hear me?
Yes. Sorry.
Yes.
All right. Great. Thanks so much. So Gavin, from what I'm intuiting, right, you're saying that there's no critical window exactly with this concept of Syngap and brain function, right? So, and I know drug development, because I'm involved in it myself, takes a really long time. So just as a clarification, is there an expectation if we have kids who are, 7, 8, 9, 10, somewhere around there, and we're talking about 10 years out, there being a potential solution that helps with that regulation of the Syngap protein, that even those older patients would be helped? And I'm sure there are other parents who have, children who are 20 years old right now, or et cetera. Can I just, from a simplistic perspective like mine, is that a correct interpretation?
Yes and no. So these are not easy These are not easy questions to answer. Especially when you're talking to families. So I have to be careful exactly what I say. I mean, I'm telling you what I think, but I have to be careful how I phrase things. So, my original paper, this big splash paper I had in 2012, the first Syngap modeling paper, explicitly states a Syngap 1 critical period. Okay. And what that means is that there are functions of Syngap that are tied to developmental periods that we think are in mice, and we think that those periods are in humans, and no one knows exactly when they are in humans and when they end, but they're probably present. And so that made a lot of parents sad, frankly, right? So what I had done, and I thought a lot about it, and talked to people, other scientists, and many of us came up with the kind of idea at the same time, is that these proteins, these genes are disease genes for a reason. There are very few protein genes like Syngap. It's just a handful of them, okay? Like there's a famous paper by the Buxbaum group that came out this year in Cell, the Satterstrom paper. And in that paper, they outline the genes that pretty much cause a form of autism, right? And Syngap is the top on the list, the top 3. And there's maybe only a dozen to 2 dozen proteins that have that kind of power on brain development, okay? And so why out of the 20,000 genes, why is Syngap special? And so the way that I reason why Syngap is so special is because it encodes a protein that's very unique, that does lots of different jobs. There isn't just one Syngap protein. There are actually a dozen Syngap proteins that are called isoforms that are made from the same gene, but they have different structures within them. And so the idea is that this is the emphasis I had from the first slide. So if you just stay with me, you'll see where I'm going with this. Is that there's more than one function of the Syngap 1 gene. Some of those functions direct developmental processes that probably do have critical periods, and that there probably is a window, that could close. I mean, that may be what's happening. I can't say that for sure, but the really important thing is that there are very clearly functions of Syngap that are not linked explicitly to development, meaning that at least in a mouse, there are functions of the Syngap 1 protein that the mouse needs for its whole life to be able to change its brain with experience. The most fundamental thing the brain does after it develops is to change with experience. So you can keep a running, you have a running chart recorder of experiences, right? And you learn and you remember, right? That's who we are, right? Is our brain records our experiences, and it does that by physically changing. And Syngap is instrumental in that process. And a lot of that function can be put back into the mouse brain when we put the protein back in. So we're very excited by that. And we've seen it time and time again. We have papers that are going to be coming out that look in different ways. And we see this over and over again. And so I guess the best way to think about this is that there are windows and they may close, but there are other, and maybe those will be restored, maybe they won't, but there are probably other functions of Syngap that relate to particular phenotypes in the kids that can be restored, maybe even in adulthood. And I should say there are other genetic disorders that are related to Syngap. So Phelan-McDermott syndrome driven by SHANK3 and Guo-Ping Feng has seen essentially the exact same principle with SHANK3, where SHANK3 will cause developmental insults, but there are functions of SHANK3 that are very clearly associated, that are not associated with development and are what are called retained throughout life, meaning that if you put SHANK3 protein back in, you also would improve brain function. So this isn't a Syngap 1 special principle. I think this is generally the case for genes that cause neurodevelopmental disorders.
Hey,
does that answer your question?
Yeah, it does. And just to add color, not to take too much time, my Syngap kid happens to be a twin, And, what I find fascinating about him is that, at birth, I wouldn't have noticed there was necessarily a difference. And that, as my children have grown, now, of course, they're very different people. Their paths have been so different, but my Syngap kid can remember certain things. I mean, it's years earlier when he was little. He'll remember when we go back to a location, he gets things. But yet I can hammer through, a math lesson or discipline lesson, and he just does not get it no matter how many times you do. So it's interesting when you were talking about the concept of memory and critical periods and development as they go forward, what is sticking with them versus what is not. And so I was curious. So it does, as best as we know, science, right?
Yeah, exactly.
Hey, Gavin, the questions are piling in. So I'm going to give you like 30 seconds to read through the chat. You can read the past 5 questions quickly. And I just want to make 2 points. You alluded in the different developmental, different druggable targets and the different stages at which we can attack the problem of haploinsufficiency. So you've been encouraging us to fund or to look at other researchers who help us better understand that. And I want to just tease the audience a little bit because we have 50 parents on. SRF is going to be doing a fundraiser soonish for a new grant to look at exactly that. So we have been in conversation with Gavin and others, and we're grateful for his time and his guidance. And we are looking to better understand Syngap. And I want to make one more point, Gavin, and I'm hoping you'll agree with me, but you might not. After spending 2 years in the rare disease space, my observation is, as you've said, we have a lot to learn, but compared to a lot of other genes, quite frankly, and I'm not trying to be flattering to you here, you know that I'm not like that. We're really lucky to have you and Rick and Jimmy, and I could go on down the list. Like relative to other genes, we have a good base. And while there's a ton we don't know, parents, true or false, parents should be hopeful that we will make some progress in our kids' lives, because time is brain, based on the stable of strong scientists we have. Would you agree with that? Or
yeah, and I can. So I read through the chat really quickly, and there's something in there that I should address. There are very good questions in there. Several. There's one topic we should address. But back to your point, Mike, I agree with you. And so it isn't. I wouldn't say it's luck. It is a reality that there are fantastic researchers in the Syngap1 space, and I can tell you that more and more are being recruited every day. I mean, I've personally recruited 4 or 5 different people into Syngap1 space just by scientific conversations and interactions. And it's not because I'm some master manipulator, right? It's because the reason why Syngap has great researchers associated with is because it's an interesting gene, right? It does things. It's a special. So, it's a famous synapse protein, dendritic spine protein, postsynaptic, there's hundreds and hundreds of those proteins, right? And most of them actually have, duplicated copies of them, right? So there's, I'm sure you've heard of PSD95 and all these other, there's 4 proteins like PSD95 in the synapse, right? There's PSD95 and SAP102 and PSD93, right? And CHAPSIN110, they're very similar to each other. There's no protein like Syngap in the synapse. So it's kind of unique in its function. And because of that uniqueness, and because of the clear effects that you have when you regulate it in cells, in neurons, or in the mouse, the mouse is a pretty amazing mouse. There's lots of phenotypes to study that are unlike other mouse models. You pull people in, and you publish good work that it's interesting and inspires people, and other people want to work in that space. And so historically, genetic disorders of the brain have been funneled into a couple of different genes, Fragile X and the RASopathies, for good reason. Those were discovered years ago, and they developed robust research enterprises around them. But the era of genome sequencing has led us to genes like Syngap that can only be discovered by sequencing. And that was just 10 years ago. So these other genetic disorders had 2 decades head start. And so in 10 years, in 7 years, we've gone from essentially 0 disease-related research in Syngap to tons of papers, and it's growing exponentially. So I think if one of the points of this talk is hope, then I think you should all be very hopeful, because What we know now is a fraction of what we'll know in a few years. And in a decade, I can't even imagine. We may have solved most of the major problems of Syngap, and then it just comes down to finding the therapeutics. That's how I view it.
Awesome. I'll let you take those questions now if you want to plow through the chat.
Yeah, so there's this discussion, questions about Syngap toxicity. So it's a paramount question. So if so much effort, it's just logic, right? So if so much effort is put into strategies to raise Syngap protein, because the accepted cause of this disease for 80% of patients it is low Syngap protein. What happens if your therapy works too good and raises Syngap protein beyond what the cell normally sees, right? And so that worries people like me and other people and other scientists, and it should worry you, because every drug that works in a disease is a poison if it works too well, right? It's anytime you develop any type of therapy, you want to understand what's called a therapeutic index. And so you want to understand how much is needed to give you relief, and how much of that drug then leads to toxicity and why. And so we don't understand if Syngap protein can be toxic. Those experiments haven't been done. And so we are creating tools to study that question. And so I can tell you that in cellular systems, in vitro systems where you grow real neurons and you express too much Syngap, the neurons don't like it, and neurons will die. That does not mean that expressing too much Syngap in a brain will cause brain dysfunction, but it makes us wonder and worry. And so the only way to really test this is to engineer systems, tools in animal models first that can allow you to tune levels of Syngap. Ideally in a Syngap 1 model where you have too little Syngap, half Syngap. And then what you want to be able to do is to tune up Syngap to where you get back to 100%, which is the therapeutic, which is a wild type situation or a normal Syngap level. But then you want to push it beyond that, right? And you want to go beyond to say, what happens if I make 2 times as much Syngap as I wanted? Does the brain like that? Does the brain like that more? Maybe if too little Syngap causes poor brain function, maybe more Syngap increases brain function. Maybe it's a cognitive enhancer, right? Maybe we want our military and our pilots and our college students to take this drug. I don't know. Maybe it's poison. Maybe it's horrible for the brain. And so we have to ask these questions, and we have to do it. And so it sounds easy, and it sounds easy, and it is easy to talk about, but it's not so easy to do this in practice. And so we're creating these tools, and we're putting the effort into doing it. And we have our first revision 1 tool, a mouse, being delivered, we hope, this month. And so we're going to start testing it out, and hopefully revision 1 is enough to answer these questions, but more often than not, you need revision 2 or revision 3. So we'll see. So this is one of the answers that I'll be able to hopefully give the community, in a year or two, is that we understand what happens when you make more Syngap than you want. And hopefully the answer is it makes brains better, but we'll see. Hopefully that was Hans's question and a few other people. So hopefully that answered your question.
Are there the other one, the few questions Marta and Pavel both sort of alluded to this, is that, hey, when we rescue the mouse, what phenotypes will be affected, behavior, sleep, etc. I realize that's I think that's pretty hard to answer, but you want to take a shot at your thought on that?
Well, we looked at some, and so we're discovering new phenotypes all the time, and a lot of that work is driven by interactions with your children, right? So we're inspired to understand brain dysfunction and particular aspects of brain dysfunction by talking to you about your children, right? Because Syngap 1 sounds like it has a lot of similarities with other disorders, but at the end of the day, if you really look into it, it's unique. And so your kids have unique problems associated with Syngap 1 haploinsufficiency. and some of them share many of these problems, right? And so the ones that where everybody shares, we want to look into and see if we can model that in an animal to understand, are those the kind of phenotypes that can be fixed, and when can they be not fixed? Can they be improved, and when can they be improved? And so memory and learning is obviously something that people talk about a lot as a problem in SYNGAP1 disorders. And so we had a paper in eLife a couple of years ago where we show that you know, important parts of learning, you know, where you are and where you've been and am I there again? Those types of learning and memory seems to come back in adult, right? So markers of epilepsy, so seizures that happen in mice because they have too little SYNGAP, those behaviors, those convulsions go away when we put SYNGAP back in the adult animal. And that's interesting because that suggests that the epilepsy or the mechanisms that lead to convulsion or epilepsy aren't necessarily locked in from an abnormally developed brain. It may be that those mechanisms are there all the time and they're waiting to receive SYNGAP protein to then correct themselves. That's one experiment that worked. It doesn't mean that epilepsy will be cured if we can put SYNGAP back in an adult, but it's certainly suggestive, and that's exciting. So memory, learning and memory, and measures of seizure in animal models appear to be fixable to some extent after development. So I think that's promising.
I have one more question, and then if anyone else has something else, please message me. So, you know, I always have to throw in an advertisement. The next speaker in 2 weeks is Dr. Shilpa Kadam from KKI. She studies EEGs and sleep. So for people who have questions around sleep, I love Gavin, of course. I also love Shilpa. They're both a pleasure to talk to, and don't miss that one. But the question, Gavin, and I've put this to both you and Rick, when is there going to be something I can put in my kid is the question I ask everybody every day. And the answer from the ASO people is, I don't know. Before COVID, optimistically, I would have said a few years. Now maybe it's a few years plus 1 or 2. And then the $1 million question that was part 2 of Hans's question is, Gavin, you have this amazing drug library. You're doing all this amazing work. When are you going to have an answer? And because of the volume and the scale and scope of what you're doing and the speed of science, like that answer is can you help? I mean, we've talked about this privately, but can you share whatever you want to say publicly around how parents should frame their hopes, assuming we continue to support and encourage science, around when there might be actual therapeutics we can dose our children with?
Yeah, you know, and you know me, I try to stay grounded, and I try to give. I'm not an egomaniac. I try the best I can. I think I'm a good scientist, and I think I can make a difference. But at the same time, I'm grounded, and I try to keep a reasonable outlook. ASOs, if they work, yeah, it's going to be exactly what industry says. Because you know, there's been the development timeline for nusinersen is a known quantity. They know how long it took. The successes that have been publicly demonstrated for ASOs related to Dravet models, that went really fast, right? So that's one of the benefits of ASOs is that if that mechanism works in a gene, it can be fast. You know, the question is, what if it doesn't work? What is plan B? And so that, you know, there really is no plan B. I mean, there's another type of ASO that might work for a type of Syngap1 transcript that might be inhibitory, but that's, you know, that's a very defined biology. If it works, it's exciting. If it doesn't, there's no plan B. And so what I try to, and this is where I try to stay grounded, is that the screening that we're doing is to uncover. Really, it's a way to uncover other ways, like what are all of the ways we can regulate SYNGAP protein? Because this is what we got to do. And so my unbiased screening system, because I'm at Scripps, I can throw 200,000 compounds at this system, and I can what's called back translate the mechanisms that regulate SYNGAP protein. Okay, and so that's going to lead to a whole bunch of biology projects, and that's going to lead to a better understanding of how can we raise SYNGAP protein. That's going to lead, we think, to plan B, plan C, plan D, plan E. Like, I'm not going to develop those drugs. I'm going to tell drug companies and pharmaceutical companies, these are what you should be looking at. You should be making drugs for these, right? So I'm trying to elucidate ways to do this, okay? If I had $20 million, if someone dropped $20 million on my door, on scripts, you know, in front of my office, it would be accelerated because it's a manpower issue. I've been lucky to get NIH funding for this, just because this platform that I developed, you can use for any single gene disorder, right? It's a discovery tool for regulation of any gene that causes disease through too little expression. So the NIH likes that because they're like, if it works for Syngap, it might work for, you know, other single gene disorders. So I've been lucky in that way, but because of that, I have to kind of always keep one eye on how flexible is this for other systems. So I can't go full blast on just Syngap1 because it's more of a general tool, which is where, you know, if I was lucky enough to get, you know, some large, you know, gift from somebody, I could put people on it and they would be able to work just on the Syngap1 stuff. And I'm not asking for money from anyone. I'm just saying at the end of the day, there are some projects that are just, we don't know enough, we need time, we need to do research. And there are some projects like screening where it's a manpower problem. The bottleneck is manpower.
Well, let me do that for you. I am asking for money. Like that's why we have a postdoc in your lab. And if we as parents could raise more, we would have more postdocs in your lab. So if anyone wants to give money, 100% goes to the labs. But I was trying to push you to the hard part, Gavin, which was, if we hope for ASOs in 3 to 5 years, which is the number people have shared with me, the scale and scope of your work, you're not, because parents say, when's Gavin going to be done? And I don't think they understand that we're talking years, right? So this
is a biology project. This isn't a drug. So okay. So I hear what you're saying. So there are ways, so Scripps has unique libraries that other people don't have. Okay. Right. And so we have the world's largest repurposing library, right? We have this 12,000 compound library of every single drug-like compound that is safe in people, right? It contains all of these compounds that failed in clinical trials but are safe in people. And so we're screening that stuff now. And if we find compounds that get validated and erase Syngap protein, those can be tried soon, you know, right? That's why repurposing is so exciting, is because the avenue to trying them in the kids is fast, you know, but the chances of that working are low, but it might work. And so if that doesn't work, then we have to go the longer route, which is to try to find probes that basically, those probes will tell us about what are druggable pathways that raise Syngap. And it could take 10 years, right? I mean, I can't put a timeline on it. I can say the repurposing screen that we were doing it and then COVID hit. I'm not exaggerating. We got the libraries from La Jolla in February, and I ordered $15,000 worth of reagent to screen that, and it would have been done now. It would have been done by now. But COVID, we were shut down for 3 months. So now, you know, we're hoping to start that screening in September. That's how bad COVID screws everything up, because you have to shut everything down and then restart the machine, and it takes a lot of time. I'm not complaining. I'm just saying it's not as simple as just taking some meat out of the freezer and thawing it and cooking a steak. You gotta, like, you know, mice need to breed, you know, reagents have to be ordered, like infrastructure has to be rebuilt, that kind of thing.
None of us think what you're doing is easy, and we're grateful you're doing it. We're over time, but we still have 40 parents hanging on. There were 2 questions that came in. I don't know if you want to jump into these because they're big. One of them is, any insights into geno-pheno correlation? It's like a $2 million question. And then sort of a second order question about penetration of brain with ASO, which I think gets us into the complexities of delivery for ASOs. You want to opine on those?
Yeah, the first one I'll take first because it's easy, is that I can't remember what's publicly available and what's secrets that I've been told, so I can't comment. Yeah. All I can say is that it's a good question, and I think largely it's still unknown. I think there's some data published in Human that says that they penetrate human brain pretty well. And I know I can say this because of my knowledge of Syngap is, you know, the human brain is huge volumetric mass. And so Syngap is expressed very heavily on the very outer part, right? Yep. So your brain is floating in a sea of CSF, and those ASOs will be swimming on the outside and are going to get in from the outside in and from the inside out. And so Syngap is on that outside. So that's promising. But there needs to be more work on that. And that's a big unknown. The geno-pheno correlation, it's a great question. So the big elephant of my talk is that people, especially for people that have missense kids, is that there's been so much work done on the what are called the clearly pathogenic, so the nonsense and the frame shifts and the deletions, is because we know we're pretty 90% confident that the mechanism is shared amongst all those kids, which is low SYNGAP. And so one of the things that we're going to start doing is we're going to start doing a consortium with some other people, Jimmy Holder and some German scientists at Max Planck, to start doing geno-pheno for the missense. And so some of you may be aware of that, and we have a worldwide effort at trying to collect genetic reports from kids, and we want all the reports. We want the nonsense, we want the missense, and we're building maps of all of these variants, especially the unpublished ones. And so what we're going to do is then do a systematic geno-pheno of missense variants. Especially, we're going to look for areas in the gene that cluster. We'll make human cell lines, human iPSCs. We'll make mouse models, and we're going to try to understand. And we're going to try to answer this question that I'm just going to pose. If my child has a missense variant and they have this phenotype, do I think that my child would be a candidate for SYNGAP raising therapies? That's the question we want to answer, because that answer is difficult. That's hard. We can't give that answer to parents now that have missense kids. But we think by doing these maps and comparing the effect of SYNGAP, the effect of variant on SYNGAP mixed with the clinical phenotype, we think we'll be able to give that answer after this project's over. And this is another kind of 5-year window project. So we're working on it. So hopefully that's a little bit of news.
Yeah, it's exciting news, and we're excited to support it. The questions are flying in, so I don't know if you want to take a quick look. So
I can stay for a while longer. It's not a problem.
Let's go through JJ's really quick. Getting back to basic biology, what needs to be done to get back to understanding the very basic functions of how SYNGAP is transcribed, translated, and regulated by the cell? And getting back to the range of phenotypes, is it haploinsufficiency of isoforms of a SYNGAP protein, and how do we alter that? So I want to pile on. Could you just elaborate a little bit for people who haven't heard about isoforms and just sum up, like, there's a big one, there's other ones, and what we know and what we're still learning?
So isoforms are important, but they muddy the waters in terms of complexity. So isoforms are confusing for neurobiologists, so they're going to be really confusing for everybody else. And so I'm going to try, can I share my screen again?
Yeah, go for it.
Because I have something on here. It's not data, don't worry. Can you see that? No, not yet. Share.
There you go.
Okay. And so it was my very first slide, I believe. Yeah. So let me zoom. Okay. Okay. So you can see this is the SYNGAP1 gene. Okay. And so there's one SYNGAP1 gene. All right. But there's no one SYNGAP protein. Okay. There are many, we call them the SYNGAPs. And so, and the reason why there are multiple SYNGAPs is that there were called alternatively spliced exons. And so you don't need to know that, other than when you make a SYNGAP1 message, you mix and match different modules and you put them in like building blocks. And then they can encode each one, and you can see my cursor, I assume. So like these are present in some, but not all SYNGAP1 messages. So when this is present here, you end up with a SYNGAP protein that has either this protein end or this protein end, right? If this isn't included, you end up with this protein end or this protein end. There's been some research by my lab and also Rick's lab that shows that these parts of the protein that are mixed and matched within all of the SYNGAPs impart different functions of the SYNGAP protein. And so that's one of the ways that we think that SYNGAP is a disease gene, is because it encodes proteins that actually have different functions from the same gene. Now, what does that mean for therapy? So that's interesting. That's one of the reasons why people think SYNGAP's cool. Biologists think SYNGAP is cool, but what does that mean for you all? And so what that means is that a therapeutic question is do I need to raise all the SYNGAP1 isoforms? Well, I guess you can ask it. A more basic question is does my child have a disorder because they lost one SYNGAP isoform or all the SYNGAP1 isoforms? And so that's important, because which ones do I need to put back? Ideally, we don't know. And so the ideal thing would be to put them all back in like nature intended. And so hopefully the ASO therapies can do that, because that question's answered. Now this there's another side question is that if ASOs don't work, a viable plan B or C, no one's really brought it up, could be gene therapy, right? So you could put a functional copy of SYNGAP1 back in the brain. That's a little bit further off because delivery of large messages like SYNGAP are tricky now, and there are companies that are working on that to try to solve that problem. And they're also trying to take a functional copy of a gene and be able to regulate its expression so you don't give too much or too little. But that's going to be some years off. But that's possible. But the question is that when you do viral delivery, you have to pick one of these isoforms. You can't put the whole SYNGAP1 gene back in. You have to pick one of these isoforms and you use what's called a cDNA. So it's very, very important to know which one of these is sufficient to bring back function in the brain that is related to the phenotypes that we all care about, right? And that you all care about. And so that mouse that I have that's studying toxicity, it also answers, we think a clever mouse because it also answers the question that we're getting at now, which is which isoform is sufficient in a gene therapy setting to bring back these isoforms. And I can tell you that it's this alpha-1 isoform that we think is very, very important. And we're testing that.
You just said something that made my brain wake up a little bit. You said when you do viral delivery, we have to choose an isoform. Are you referring there to ASOs or AAVs or both?
No, so like AAV, but AAVs won't work for SYNGAP. It's too big. But but something like an AAV where you package a SYNGAP message into a viral vector and then inject it into the body, and then it goes and what's called transduces. It enters the brain cells and then inserts a copy of SYNGAP1 in there that's functional. And then from that copy of SYNGAP1, it makes protein. So that's another way to make more protein.
But that's not an ASO. An ASO would be taking down an existing regulatory mechanism, right?
An ASO would be an artificial nucleic acid, so like something that looks like a RNA or DNA.
Right.
And what it will do is, you can see my screen still, right? So what it'll do is, you have haploinsufficiency, so you have half the mRNA. What an ASO will do is bind somewhere in this process to the mRNA that's still there and basically tricks it into being more efficient.
Yeah.
So, right?
Yep,
it'd be like pumping up your tire twice as much, right? You're taking whatever's there and making it work twice as well. Whereas a gene therapy would insert a new copy of SYNGAP1.
So now Hans is getting going and he's asking about prime base editing. So can I just, for the sake of time, tell you my high level answer when people start asking me about gene therapy and base editing, and you can correct it. It's a little selfish, but I want to keep it simple. When I'm talking to parents, I say, look, there are ASOs in development right now. There's depending on how you count, let's just say a few plus, and those might be available in 3 to 5 years. Gavin is doing this massive drug repurposing that might come around in about 5 years. Then there's this, we're not a good candidate for current gene therapy, AAV9, but next gen gene therapy maybe, and prime base editing maybe. But the way I think about the world, the next stop on this train is ASOs, and then shortly after repurposed drugs, and then in the future as the technique gets better refined and generally tested out on Dravet first for a variety of reasons, we can look forward to other viral vectors and or prime base editing. Is that directionally correct?
Yes, my gut tells me yes. So I can't add anything to what you said. It's true. That is the time frame. But my knowledge of the science, my gut will tell me at the rate at which everything is accelerating, I think the future looks incredibly bright for viral therapy. So there you buys your back. I think the future looks very bright for viral therapies, as long as there isn't some unfortunate roadblock along the way that happened in the 90s, at UPenn. So I think that the technologies are there. And I know that there are very smart people in pharma that have started venture funds and startup companies specifically for this problem. So I think it's very promising, but it's going to take, there's some technical hurdles to overcome. But when those are overcome, I think it's going to be very promising.
Awesome. Can I throw one more question at you and then we can let this wrap up? It's
Sure.
one Martha just asked, and again, I'm going to just opine for a second. people come at me when I talk about ASOs and they're like, what about gene therapy? And I say, A, we're too big for the gene therapy vector AAV9, but B, gene therapy is one and done, whereas ASOs you can titrate. So we are and we should continue to be focusing on ASOs. Could you just elaborate on why a viral vector is you get a one-time shot versus ASOs are repetitive?
Well, currently viruses are a one shot, and that's one of their, it's a drawback for like a non-lethal disorder, right? So like, you know, one shot is fine if the alternative is not non-survival, right? But so that's not the case for Syngap and most genetic disorders. So, you know, you could do more harm than good with when you can't regulate the expression of Syngap if you put it in, right? That's dangerous because we don't know if it's toxic or not, right? So there's no way you can do that. For viruses, that's one of the hurdles that people are trying to solve in industry right now, which is put kill switches in. So there's an investigator at Scripps that has had nice papers in Nature Biotechnology showing you can put kill switches in viral vectors. So if it starts to become toxic, you can just kill it, turn it off. And then people are trying to take the kill switches and turn them into volume controls, if you will, right? So, you know, you want to be able to turn up and down the volume of how much protein you make off that artificial gene, right? That's what you want to do. And that's what my mouse models can do in theory. It would be awesome to have a translational strategy that you can do that, because if you could, and you could put any gene you wanted in there, you'd have in theory cures for, you know, most of these genetic disorders, but that's some time off. ASOs are different. You have to inject it. It's a 20 base pair artificial RNA, DNA, depends on how you look at it. And so what it does is it goes in and binds to Syngap mRNAs, pre-mRNAs, and tricks them into doing things. So it's like a drug. Like it's just a drug that you can't take by pill. You have to inject it currently into the intrathecal space because it's big and it doesn't get across the blood-brain barrier. So you have to inject it kind of into the brain. That's why they go in through ports or through the spinal cord.
Awesome. I'm very grateful, Gavin, for how generous you've been with your time. And I think all the parents on this call will agree. I'm sure the questions will pour in, and we might send you a sum up of the burning issues. But on behalf of all the parents on this call, thank you so very much for making yourself available. This was awesome.
It's my pleasure. It's good to see everyone. And hopefully I'll see some of you, many of you in person soon.
Yeah, I think the Floridians are going to have a hard time staying away from your lab. I keep telling Sydney and Vicky to go see you. And you can see the chat is blowing up with gratitude. So really appreciate it, guys.
It's my pleasure, genuinely. And like I said, I hope to meet all of you in person someday.
Yeah, us too.
All right.
Take care, everybody. Thank you.
Cheers.
Just want to remind you, in 2 weeks, we'll also have Shilpa talking on July 2nd. This is on this SRF Facebook page. You can register with that link at the bottom. She'll be talking about some of her specifically SYNGAP1 epilepsy research. And then also Helen Badeup will be 2 weeks after that. And then a reminder that we have 5 days left on this Be Kind campaign. So if you want to get shirts, again, the link's on our Facebook, but SRF Kindness Off Bonfire. SRF does a weekly parents meetup, which is always fun and generally useful. I took the least flattering picture possible of everybody in this one, but they're good meetings. And then just a reminder, we have the Syngap Research Fund board is now up to 12 families, and we're always looking for people who want to help. So if you are, if you have the time to do more and you want to be involved, reach out. SRF is pretty much everywhere. We're on Facebook, Instagram, LinkedIn, Twitter, and syngapresearchfund.org is a good website to have handy.